The Atlantic bluefin tuna is a large predatory fish under extreme fishing pressure. In order for effective management of the Atlantic bluefin to take place, it is vital to understand the dynamics of its populations. This map shows positions of electronically tagged Atlantic bluefin. The data show that there are at least two separate populations of Atlantic bluefin tuna. One population spawns in the Gulf of Mexico, which is represented in the top map by orange circles. The other population spawns in the Mediterranean Sea and is represented by white circles in the bottom map. The east coast of North America, outlined in red, is a mixing zone for these fish. Currently, it is difficult to identify the individuals from one population or another in this mixing zone, which further complicates the challenge of ensuring a future for these giant fish. One way to determine the percentage of fish from each population in the mixing zone would be to tag the fish and trace them for years until they enter one of the two known spawning grounds. Though this method has been successful for a number of fish, it takes years and would be very costly to conduct on large sample sizes. Due to the differences in chemical composition of seawater in each breeding zone, microconstituent analysis of fish's otoliths or inner ears is also a promising way to determine from which spawning ground they have come. Otoliths also come with a drawback, however. The tiny inner ear bones must be dissected from the often discarded heads of caught fish. Genetics is a third way of potentially identifying from which population an Atlantic bluefin has come. Of the three methods mentioned, sampling for genetics is the simplest. Only a small piece of tissue is required, thus, a fish can be caught and released alive. Otherwise, even a piece of a fillet from a seafood market would provide sufficient DNA for analysis. Okay, so once the DNA from an Atlantic bluefin has been analyzed, there, written in the DNA code, is which spawning ground it has come from, correct? Huh, if only it were so simple. This is where building DNA profiles for each population comes in. Tagging shows that adults return to the same breeding grounds to spawn each year. This homing behavior is called spawning site fidelity. Approximately 6,400 miles separate the Mediterranean Sea and the Gulf of Mexico. Thus, it is not surprising that recent genetic studies show that there are genetic differences between the populations that breed in these separate spawning basins. Yet, because of the degree of conservation of genes in these fine-tuned predatory fish, the genetic differences found are very slight and were not in fact detected until sampling methods were refined. Analysis of microsatellites, or short repeating sequences of DNA, has also allowed for a higher resolution look at genetic differences. Using samples of fish of known origin, we are currently establishing genetic profiles for the Gulf of Mexico and Mediterranean populations using 30 microsatellites, which serve as DNA markers. Hopefully, fish of unidentified origin can eventually be matched to either the Gulf or Mediterranean profile and assigned to that population. So, why is this important? Currently, fisheries do not consider genetic populations or groups of interbreeding individuals, only geographic clumpings. Thus, the east coast of North America, despite being composed of genetically different individuals that are not necessarily exchanging genes through breeding, are managed as one. The ability to assign fish would help us answer questions such as, what is the percent composition of each population in mixed zones? And, does this percentage change from year to year? Better understanding trends such as these will give insight into yearly variations in spawning success rates for each population, and population abundances, which will hopefully lead to more specific management strategies. In addition, we as scientists cannot help but express our excitement at potentially unveiling a few more of the mysteries of this magnificent fish.